Hello students, welcome to part 4 of Control and Coordination chapter. Today I am going to discuss about a very important topic, that is action potential. We have already studied the structure of neuron. A neuron consists of dendrites, a cyton and axon. Let us see how the generation and the conduction, how it is conducted of the nerve impulse takes place. Now, bundles of axons, if they are traveling together, we will call them as a nerve. Nerves can be very long. Nerve impulse is a wave of bioelectrical or electrochemical disturbances. So, there are some electrochemical changes or electrical charge is developed. So, it is also called as bioelectrical disturbances that is passing along a neuron. It is a wave of depolarization of the membrane of the nerve cell. What is depolarization? You will get to know in few minutes. The nerve impulse travels along a neuron or across a synapse. What is a synapse? Synapse is a junction between two neurons. So a wave of impulse will travel from one neuron to another neuron through a junction or synapse, or it may occur between a neuron. Suppose this is a neuron and there is a effector muscle, any muscle is there, or neuron and a gland is there. So such type of impulses which are transmitted from neuron to muscle, neuron to gland, one neuron to another neuron, it is nothing but because of a nerve impulse, which is a electrochemical or a bioelectrical disturbance. Each neuron, now we will see it in detail, has a charged cellular membrane, and it has a voltage difference on inner and outer side of the membrane. Plasma membrane separates the extracellular fluid from the intracellular fluid. Look at this diagram. This is the cell membrane bilipid layer, which we also called as plasma membrane. This fluid which is present outside, it is the extracellular fluid. The fluid which is present inside the exoplasm or inside the neuron will be called as intracellular fluid. And what is there? There is a voltage difference on the inner as well as outer side of the membrane. Why? Because different types of ions are present. Look at the outside, that is extracellular fluid. We can also call it as ECF. Extracellular fluid, it contains a lot of sodium ions. Look at these blue color things, they are sodium ions. So many sodium ions are there. Chloride ions are there. Very few, but yeah, chloride ions are also there. And then there are lot of calcium ions. These white color structures are calcium ions. So there is lot of sodium, lot of calcium, lot of chlorine, of course a little bit of potassium also is there. Effectively, there is more of positive charge on the ECF towards the extracellular fluid, whereas on the inside of the neuron inside membrane, there is lot of potassium present in the intracellular fluid. Along with, of course, chlorides will be there. These are chloride. These black color spots are chlorides. These orange color spots, you can see they are potassium. Little bit of sodium is also there. Blue color, you can see these are sodium. So all is there, but more of potassium is there. And very important, inside there are lot of proteins which are negatively charged. As there is lot of protein, as there are lot of chlorine, the inner side, the inside or the intracellular fluid, it is negatively charged. Whereas there is more sodium outside and calcium ions also outside, so it is comparatively positively charged. So I hope you understood. Plasma membrane is a bilipid membrane which is separating the extracellular fluid and the intracellular fluid. Extracellular fluid has more of sodium, chlorine, calcium. Intracellular fluid has more of potassium, but it is negatively charged because it has lot of negatively charged protein molecules present inside. Now let us study about what is resting axon or polarized state. It's very simple, students. Let us see one by one what it is. The activation gates of sodium and potassium are closed. Look at this picture only again. This is the ECF extracellular fluid. This is the internal fluid, intracellular fluid also called as cytosol. You can see there are gates. This is a sodium gate. This is a potassium gate. This is a again a sodium gate. These gates are closed. Sodium and potassium gates which are present on the plasma membrane, these gates are closed. As these gates are closed, the extracellular fluid, it has a positive charge. Intracellular fluid, it has a negative charge. You can see in the picture, outside there are more sodium, high sodium, low potassium, low chlorine, therefore positively charged. ECF is positively charged extracellular fluid. But inside there are negative ionic organic ions. They have lot of proteins, few nucleic acids, and lot of potassium, but these proteins give them negative charges. So this potential, it is nothing but the resting potential. So how can we define a resting potential? We can say that the potential difference that is seen in a resting nerve, this is a resting nerve. At resting state, outside ECF is positively charged because of more sodium, inside is negatively charged because of more proteins and chlorides, and of course potassium, though it is positive. And this potential difference seen in a resting nerve, it is called as resting potential. And how much it is? This resting potential, if you draw a graph of time against the membrane potential, this is on y-axis we are having membrane potential, on x-axis we are having time. If we draw a graph, we see that the resting potential is minus 70 millivolts. The nerve membrane not only has leakage membranes, there are leakage membranes. The leakage membranes are there. There are some leakage membranes gates over here. They will see that the sodium is kept outside and the proteins and the potassium is kept inside. And it creates a resting potential of about minus 70 millivolts. What it is, again I tell, it is the differential permeability, differential ionic concentration, and therefore inside the potential is minus 70 millivolts. More negative ions are present inside. Now what happens? Then starts depolarization. What is depolarization? A stimulus comes. A stimulus comes. Somebody comes and gives stimulus. Suppose somebody is touching you, or you see something, or you hear something, or you smell something. So, such type of stimuli, they open the activation gates of sodium channels. Whenever a stimulus comes, look at the side B now. At A, it is still at the polarized state. Polarization state means outside positive, inside negative. But at the B region, some stimulus has come. Because of stimulus, channels are open. And as sodium channels are opening, sodium is passing inside. The sodium starts traveling from the ECF to the intracellular fluid. So influx of sodium takes place. Sodium starts traveling from the outside membrane, from the extracellular fluid to the intracellular fluid. What will happen if so much of sodium is traveling inside? Positive charges will increase inside, isn't it? And thus, what happens? The positive charge increases. And if you draw the graph, you will see that now this minus 70, which was initially inside, it starts growing. It reaches minus 55. More and more sodium will come in. This is called as the threshold phase. When it is minus 55 millivolts inside the potential, then we call it as a threshold phase. Again, more and more sodium starts coming inside. And one more thing is done. When sodium is coming inside, the potassium channels are closed. They are closed. They are not allowing the potassium to go outside, but they also maintain potassium inside. So now sodium is also inside, potassium is also inside. The positive charge increases till plus 30 millivolts. And this change in the ionic potential in the intracellular fluid, which now suddenly reaches from minus 70 millivolts to my plus 30 millivolts, see, it is reaching, it is crossing above zero. This is called as depolarization. So depolarization occurs. You can see in this case only the sodium gates are, you can see they are open. This is the sodium gate, it is open, and potassium gate is closed. What is the sodium gate? When they're opening, they are pushing the sodium ions, pumping the sodium ions inside. Potassium gates are closed, so they are also pushing the, keeping the potassium ions inside. And therefore positive charges increases now inside the cytosol. Outside becomes negative and inside becomes positive. What is this? Depolarization. Stimulus is opening the sodium channels, influx occurs, action potential is triggered. This is called as action potential, change in the ionic potential. And you can see that outside it has become negative now because of more of chlorine ions outside, inside has become positive because of more of sodium. This is called as depolarization. And what is the potential difference now? It is plus 30 millivolts. But as soon as it reaches plus 30 millivolts, then the process of repolarization begins. What happens at repolarization? Now the potassium gates are opened and sodium gates are closed. See, the sodium gates, they are closed. These are the sodium gates which are closed. Let us look at the diagram. This is the potassium gate. Potassium channel opens. As potassium channel opens, the potassium are effluxed outside. As the potassium effluxes outside, continuous potassium is put outside. The inside starts again becoming negative, isn't it? Sodium channels stop, they close, and therefore the influx of sodium is also stopped. Sodium also stays outside. As sodium stayed outside, potassium also is pumped outside. What is happening? Outside is becoming positive and inside is becoming negative. This is called as repolarization. But this repolarization starts after the action potential reaches plus 30 millivolts. And once the repolarization starts, it will go, the value will again reach to minus 55 millivolts. Then it will reach this minus 55 millivolts. Now it is called as the hyperpolarization or refractory period. Again, continuous potassium is pumped out, sodium is maintained out. Again, the inner side will become more and more negative, reaching to the minus 70, that is resting potential. Sometimes reaches to minus 90 also. This, when it is reaching minus 90 millivolts, it is called as hyperpolarization. Okay, minus 90 is hyperpolarization. But then it is finally maintained again to minus 70 millivolts, which was initial, like this initial one, that is the resting potential. Thus it is called as repolarization. Okay, I hope you are understanding. Initially, it was resting potential, outside was positive, inside negative. Then what happened? Sodium channels open, sodium starts traveling inside, the potential difference starts increasing, it reaches plus 30. As soon as it reaches plus 30 millivolts, now sodium gates will close. They will retain the sodium outside, potassium channel opens, potassium is pushed outside, outside again becomes positive, inside cytosol again begin negative. Thus repolarization occurs. You can see, so in this way the wave of polarization and repolarization is continuously maintained by these gated channels. These channels which you saw in this picture, these are sodium gated channels, these are potassium gated channels. They close and open as needed. And again, in this first phase, you can see this is a polarized membrane. Outside is positive, inside is negative. Why negative? This is also called as a resting axon. No stimulus has come because outside there is more sodium, inside there is more proteins or negative ions. Stimulus came, this region, it depolarized. And then the wave of depolarization passes from one end to the other end. So suppose this is A, this is B. This has A has now become depolarized. This is B, and this suppose we will name as X. X becomes repolarized. So wave of polarization, depolarization, and repolarization slowly passes from one region of the axon to the other region, and this way the electrical impulse is conducted by changes in the ionic, what we'll say, minerals. Now, sodium and potassium pumps operates actively by use of a carrier. Always remember, students, for three sodium ions, two three sodium pumps out, two potassiums are pumped in. So a balance is maintained. If three sodium are pumped out, then two potassium will be pumped in. And when you should always remember this, I hope you have understood the concept of resting potential, polarization, depolarization, and repolarization. Also, one more important thing you should remember, in medullated nerve fibers, the insulating fatty myelin sheath, it prevents the flow of ions between the exoplasm and ESF. So the transport pump and gated channels can operate only at the nodes of Ranvier. In case of myelinated neuron, it takes place only at, this process occurs only at the nodes of Ranvier. So this type of conduction is saltatory conduction, which is very fast. It will take place at a rate of 120 milli per second. This is the rate. It is very much faster than the continuous conduction in non-myelinated fiber. The ratio is 50 is to 1. So it is 50 times faster conduction in myelinated neurons as compared to non-myelinated neuron. So students, I hope you have understood the concept. Now in the next video, we will discuss about what is synapse and what are the different types of synapse. Thank you.